So we'll quickly go over parts of a neuron. We've talked about this a bunch, so it's probably a review. The cell body is known as the soma. Within the soma, we have the nucleus. The plasma membrane specifically is called, the plasma membrane encloses the cytoplasm, which is specifically called the perikaryon. And inside the cell body, something that we want to focus on in lab here are something called the chromatophilic substance, or the Nissl bodies. And that's basically just these little tiny dots that kind of, they stain a dark color when you look at them under the microscope, and they're made up of ribosomes, so like free ribosomes and bound ribosomes. The dendrites are those short unmyelinated processes that come off the cell body, and the axon is going to be the long process emanating from the cell body. So in the ones we're going to be identifying, we're going to be looking at mostly multipolar neurons, and so you'll have many dendrites and just one long axon that comes off. And then most of the axons we'll look at are myelinated. The place where the axon attaches to the cell body is called the axon hillock. The cytoplasm inside the axon is called the axoplasm. A lot of these words you don't need to know for class. It's just kind of FYI. Make sure to look in your lab manual. You do need to know the axon hillock, the synaptic knobs and synaptic vesicles, but the other ones are more a little bit FYI. Just if you read them, you'll know what they are. Another thing you can call the synaptic knob would be the terminal bouton or the synaptic bulb. So I'll accept any of those. All right, so axons are going to be important in conducting action potentials all the way down, well, down the axon, and then when it gets to the synaptic knob, it's going to release those neurotransmitters. The cytoskeleton here, this is going to be made up of those little tiny protein elements. So the microfilaments are the smallest ones, the intermediate filaments are the medium ones, and the microtubules are largest, just like all of our normal cells. But specifically in our nerve cells, those intermediate filaments are termed neurofilaments, and they're going to kind of go together and bind together to form these bundles called neurofibrils, and that kind of gives some strength to the cell. So here, this has got a lot of the different things, the cell body, the dendrites up here, the axon hillock, and this part is the axon. And then here we have these little neurilemmacytes. So can you tell if this axon is in the peripheral nervous system or the central nervous system? Wait a second. So it's in the peripheral nervous system because this is a neurolemmacyte, and we can tell that because each of them has its own nucleus and its own neurolemma, and that's only found in the peripheral nervous system. In the central nervous system, you have one oligodendrocyte that's going to form multiple areas of myelin sheath across multiple different axons. In here, we can see these little dark staining areas in here inside the soma are going to be the chromatophilic substance. Then we have the nucleus in the middle here and the nucleolus. And then you can see this is the axon coming down. Here's the synaptic bulb, and it contains those synaptic vesicles. And this is much like what we talked about with our muscle tissue at the neuromuscular junction. So once again, rehashing myelination, this is really important. The more myelin we have, just if we have myelin in general, we are going to be increasing the velocity or the speed of that action potential. Myelin is going to be layers of the plasma membrane of those glial cells. So either in the peripheral nervous system, it's going to be a neurolemmacyte, and we can also call those a Schwann cell, or in the central nervous system, it's going to be an oligodendrocyte. And that, because there's lots of lipids, it gives a glossy white appearance. It also insulates that axon. And so remember that in the PNS, each of these neurolemmacytes has its own nucleus, and that nucleus then, so that means each of these little myelin sheath sections is going to have its own outer layer with a nucleus, and that's called the neurolemma. And that's going to be really important for helping the peripheral nervous system regenerate better than the central nervous system. So a neurolemmacyte can only myelinate around 1 millimeter of axon, so several of them are needed, a lot, for one axon. And so the gaps in between those neurolemmacytes are called the neurofibril nodes, or the nodes of Ranvier. All right, so that was our nervous tissue. Hopefully you guys have seen that before. There are a couple of good pictures of the neuron in your lab manual, as well as on the practice lab quiz online. I've got some pictures of another type of neuron you guys can look at to practice labeling. So now we're going to move on to the brain. I'm going to go through these slides here, and then I'm going to show you a little bit on APR to show you some of the things that you should be looking at. You have APR at home, you can use this. It's a great resource, especially with this online lab. So 4 major brain regions. We're going to have the cerebrum, which is made up of 2 hemispheres, and there are 5 lobes per hemisphere, the diencephalon, the brainstem, and the cerebellum. So we'll kind of start with the cerebrum and go down. On the outer surface of the cerebrum, or the outer surface of the brain, it looks a little bit wrinkled. The ridges are the gyri, and the sulci are the depressions between the ridges. So if you think of like hills and valleys, the ridges or the hills would be the gyri, and the sulci would be like the valleys. A fissure is like an extra deep sulci. Another time, so if we go towards the front of the brain, we can also call it rostral. So anterior is rostral in the brain, and posterior is caudal, which kind of means towards the tail end. Like towards the ponytail? Oh, no. Towards the tail end. And if we look here, let me pull up my Anatomy and Physiology Revealed. So here is one of the outside of the brain, and you can see that these little areas here are going to be the gyri. Let's see, I don't think specifically. So this is one of the sulci, the lateral sulcus. So the area in between the gyri are the sulci. So the gyri are the lumps, and these little guys here are the sulci. All right, so let's get back to here. So once again, this is going to be the gyri, and then the areas in between are the sulci. So we can see here that this is going to be the anterior, so this is the front or rostral, this is posterior or caudal, and the frontal lobe is in the front, so that's this part right here. Then right behind that is the parietal lobe, and there are 2 parietal lobes. They're paired, just like the bones above them. So remember, we have the frontal bone in the front, the 2 parietal bones on the side, the occipital bone in the back, the temporal bone on the side by the ears. This is the temporal lobe. So the lobes are named for the bones that overlie them. So if you know the bones of the skull, you should hopefully be able to name the lobes. There are a couple of fissures to know. So this, or sulci to know. So this right here is the central sulcus, and it's in between the frontal and the parietal lobe. And then we have the lateral sulcus here, which is going to be separating the temporal lobe from both the frontal and parietal lobes. Those 2 are on your list to know. There is also this parieto-occipital sulcus. It's not on your list to know, but it just FYI, it's going to be dividing the occipital lobe from the parietal and temporal lobes. So down here, we've got the cerebellum, which is like the little brain. This part is the brainstem, and then the diencephalon is like in the middle and the inside of the brain. So we'll get to there soon. Now from the bottom, this is looking at the brain from the bottom. So like this down here is the brain stem, right? So this is going to be more inferior. And then as we go down, the cerebrum is more superior up here. We can see the temporal lobe down here, the brain stem here. This is the pons, which is part of the brain stem, and this is the cerebellum. So you'll want to make sure to look at the brain from several different directions. This is the, if we cut the brain in half, so we're looking at about a mid-sagittal view of the brain. And so once again, this is going to be anterior or rostral, this is posterior or caudal, superior at the top, inferior at the bottom. So this is where we can find the diencephalon. And I said that diencephalon is like the older part of the brain, and it's going to be right here in the middle. And it is, we'll go through all of the different things in here. So this is the diencephalon, then we have the brainstem coming here, this is the cerebellum and the inside of the cerebellum, and then here, once again, the outer part over here is going to be the cerebrum, which we'll start with. So the brain and the spinal cord are both going to be composed of 2 different types of matter called white matter and gray matter. And gray matter is going to be made mostly of neuron cell bodies and dendrites and some unmyelinated axons. So it looks gray because there's no myelin. The cortex is the superficial region of gray matter in the cerebrum. So like the cerebral cortex, that's the outer layer. Any nuclei or nucleuses are regions of gray matter that are deeper within the brain. And so a nuclei is kind of like a group of cell bodies within the central nervous system. White matter is going to consist of myelinated axons, right? So the myelin, it has this like glossy white lipid-filled appearance. It's going to make it look white. So the white matter has myelinated axons. And in the central nervous system, they are organized in bundles called tracts. So our myelinated axons are organized in bundles called tracts. So if we can look here, this is going to be taking a frontal slice of the brain. So we're going to be going through. So we're dividing the body, and we have a superior, or excuse me, anterior and posterior parts. So if we took a slice right through, we can see that the very outside here is going to be gray matter, and that's going to be the cerebral cortex. And this is where a lot of our thought happens, right? These are all the cell bodies in the cerebral cortex. And then right in here, really deep in here, we have these cerebral nuclei. That's more gray matter that's deep in the cerebrum. And then we have a lot of white matter kind of inside. And so all this white matter here is going to be inside. This one specifically is a really important tract. This is the corpus callosum, and it's going to be connecting the right and left hemispheres of your cerebrum. So that is the one that you need to identify, but you'll be able to see it pretty well on the sagittal section. So now we're going to kind of take a step back and talk about protection and support of the brain. And this is really important because our brain is very delicate, and we need to make sure to support it. So the cranial meninges are the layers of tissues that surround the brain. Now, we talked earlier in like lab 1 about the layers of tissues that are surrounding our thoracic and abdominal organs. So we had serous membranes within those ventral cavities, the visceral and parietal serous membrane inside the ventral cavities. Well, inside the dorsal cavities, remember the posterior aspect or the dorsal cavity, that's the cranial and the spinal or vertebral column. Instead of having serous membranes, we have meninges. And so these are connective tissue layers that support the brain. They're going to also contain blood vessels that supply blood to the brain. They help contain and circulate the cerebrospinal fluid. And from deep to superficial, it's pia mater, arachnoid mater, and dura mater. So PAD, I guess, PAD. So remember, deep to superficial, pia mater, arachnoid mater, dura mater. So the pia mater is the innermost layer of the meninges, and it adheres directly to the brain surface. So you can kind of think about it a little bit like the visceral serous membrane, right? It's adhered directly to the organ, which is the brain or the spinal cord. And it's a thin layer of areolar connective tissue, and it follows all the contours of the brain. Then the next layer we have is the arachnoid matter. And so the arachnoid matter is going to be outside of the pia mater, and there's a space, and there is a space in between the pia mater and the arachnoid matter. And that space is called the subarachnoid space. So sub means below, so it's below the arachnoid. So below the arachnoid and right above the pia. And so the arachnoid trabeculae are going to extend to the pia mater through the subarachnoid space. And I'll show you a picture of this in a second. And the subarachnoid space contains cerebrospinal fluid. And so these arachnoid trabeculae are made of a web of collagen and elastic fibers, and they kind of connect the arachnoid matter to the pia mater, and then they keep that space is open, and that space is filled with cerebrospinal fluid. And it's important that we know that there's cerebrospinal fluid in there. And then the arachnoid matter lies just deep to the dura mater. And so the dura mater actually lies right on top of the arachnoid matter, and there shouldn't be very much space or any space in between them. Usually they lay right on top of each other, but there is a potential space, so they're not connected. And so this potential space is called a subdural space. So if there's bleeding that occurs into this space, it can actually open up, and you can have what's called a subdural hematoma that can get filled with blood. So before we go through, so I wanted to just show, so this part right here, this is the pia mater. It is directly adhered to the brain. It's very deep. So this little pinkish area here, the pia mater. And then remember that we said there is a space in between the pia and the arachnoid matter. So that's the space here that's filled with these little spider web looking things, right? So arachnoid, think spider. And so it looks like, mater is for like mother, so it's spider mother. This is the spider area here. So the arachnoid trabeculae look like spider webs. And so this is the subarachnoid space right here, and it's filled with these arachnoid trabeculae, and it's filled with cerebrospinal fluid. And this fluid is important. That's going to help the brain sort of float. It's going to be buoyant, and protects the brain. And now so then this guy right here is the arachnoid membrane, and then right above it is this thicker layer right here. This thicker layer here is the dura mater. And so this is going to be the dura mater, and the dura mater actually in the brain has two distinct layers. So the dura mater is thicker, right? It's the tough outer covering. And so this is going to be the meningeal layer of the dura, here. And that is going to be the part that kind of follows the brain contours, right? It's going through the brain contours. And then the outer layer here is going to be the periosteal layer of the dura mater. So it's going to be directly connected to the inner bones of the skull, and it's kind of making up the periosteum, right? Because it's also tough like the periosteum. So in some areas, these two layers of the dura mater lie right next to each other. So like right here, and they're connected. But in others, where the brain, like there's this horizontal fissure here. So in other areas, we have the meningeal layer of the dura mater that is going to be moving downward, and whereas the periosteal layer of the dura mater is going to be staying connected here. And so in this case, where these two areas diverge, there's this dural venous sinus. And so this is going to be filled with venous blood that's draining from the brain. And so in this case, the dural venous sinus here is going to be filled with this draining blood. In this, there is a name, so this one's specifically the superior sagittal sinus, but in general, just call it a dural venous sinus. And so these two layers of the dura mater here, and where they diverge, we have an opening inside it called the sinus, and this is where the venous blood the venous blood is going to drain from the brain. So that was the dura mater right here. Now, outside of the dura mater, so in between the dura mater and the skull, there's another potential space, and that would be called the epidural space. And so that contains arteries and veins, and if one of the arteries or veins breaks, then you can have an epidural hematoma. So we're going to talk a little bit more about the arachnoid villus and arachnoid granulations in just a second. So, but you can see here that sometimes the arachnoid matter and the cerebrospinal fluid is going to kind of punch up through the dura and into these superior sagittal sinuses. So these are called arachnoid villuses or arachnoid granulations, and this is where our cerebrospinal fluid gets recycled. All right, we kind of covered epidural and subdural hematomas. Both of these are in case you have blood vessels that break or when blood is going to enter into those potential spaces. The problem with these is that the blood can actually start pooling like this, and it can, because your brain, your skull can't stretch, right? It's a complete complete cover because your skull can't stretch. If you have a lot of blood that starts filling it, then it can actually put pressure on the brain. And so you can compress the brain tissue, and that could be, it could damage the brain. So the other sort of clinically relevant things are that your meninges, which are those layers we just talked about, can get infected. And when they're infected or inflamed, we call this meningitis. And this is inflammation of the meninges, and often this is caused by either a virus or a bacteria. And so usually you'll have fever, a headache, vomiting, a stiff neck, like it hurts to bring your chin down to your chest. And depending on like the type and the severity, it can actually cause brain damage if it is untreated. And so bacterial meningitis usually has more severe symptoms. Encephalitis is actually inflammation of the brain itself. And that can be really bad. Symptoms are drowsiness, fever, headache, neck pain, and it can result in death. So where the cranial dura septa here are sheets of the dura mater, the meningeal layers of the dura mater, that are actually going to go in and dive next to the brain. And this is going to be important because this helps sort of keep the brain in place. So if you look back at this picture, this says this is the falx cerebri. This is a piece, one of the cranial dura septa. This is kind of FYI, you don't need to identify them, but just know that these pieces of the meningeal layers of the dura mater are going to be important in like keeping the brain in place. And we can also see here that usually where these cranial dural septa are, that's where the meningeal layer has like separated from the periosteal layer. And so you're going to have these dural venous sinuses here. And they all have different names, but you guys don't need to know their exact names. Just know what a cranial dural septa is. Ventricles are cavities that line within the brain, and they're lined with ependymal cells. And these ependymal cells, remember we talked about in lecture, are a type of glial cell, and they help make cerebrospinal fluid. And they connect with each other and the spinal cord central canal. So I'm going to show you a picture here. These are a potential space. This is like an opening within the brain. And these openings within the brain are filled with cerebrospinal fluid. I've got some good ventricle model pictures on your lab practice quiz that you guys can practice labeling. So you guys will need to know that these two big C-shaped ventricles here are the lateral ventricles, right? So we've got a right lateral ventricle and a left lateral ventricle, and they're connected to the third ventricle by this little guy called the interventricular foramen. This guy here on midline is the third ventricle, and it has a little hole in it. This is where the interthalamic adhesion would be, where the thalamus is connecting each side. Then from here, we go through the cerebral aqueduct and down to the fourth ventricle. And the fourth ventricle is here. It's going to be around the level of the cerebellum. And then we have it's paired lateral apertures that come out to the side. So you can see them here. So these paired little lateral apertures, and they're going to connect up to the like the subarachnoid space, so that the CSF can then also circulate from the ventricles to the subarachnoid space. And then we also have a median aperture. That's a single median aperture, and then it's going to continue downward as this central canal of the spinal cord. So make sure you can identify all these areas of the ventricles on that ventricle model. So the cerebrospinal fluid is going to circulate both in the subarachnoid space as well as in the ventricles. And there's a couple of important functions for it. Buoyancy, it helps reduce the brain's weight. It helps protect with the brain with a liquid cushion. And it's also going to stabilize the environment. And so basically, remember that blood-brain barrier as well as the CSF are going to be filtering things into and out of the CSF, and specifically letting certain things into the CSF and not letting other things into the CSF. So how is the CSF formed? It's actually formed by something called the choroid plexus, which is found in each of the ventricles. There's a layer of ependymal cells along with blood capillaries within the pia mater. And so blood plasma from those blood capillaries is filtered through the capillary and then filtered through the ependymal cells. And the ependymal cells kind of modify it to make it into cerebrospinal fluid. So CSF has more sodium and chloride, less potassium, calcium, and glucose. And the ependymal cells also secrete special things to help make the CSF. So excess CSF, once it circulates through, is going to go back into those arachnoid villi. It flows into the arachnoid villi, and then it drains into the dural venous sinuses. So it takes that extra fluid back into the venous system. And arachnoid granulation is a collection of those villi. So we can see these are the lateral ventricles in here, and these are the collections of the choroid plexus. And so this is the capillary with the endothelial cells, and this guy right here, these are the ependymal cells that sit right on top of there, and they're going to be helping make that CSF. There's a good video in your virtual lab that goes through CSF production. But so here it's made in the ventricles, and then it's going to go through all the ventricles. So if it starts in the lateral ventricle, it'll go to the third and then the fourth, and then it's going to go from the fourth ventricle through those apertures and into the subarachnoid space. Then from the subarachnoid space, it'll go into the arachnoid villi and then back into the dural venous sinus to be recycled. So it goes from like the capillaries that are supplying arterial blood, and then it's going to go into this CSF and then eventually back into the venous blood in the dural venous sinus. So here we can see this is the arachnoid villus, and it's going to be kind of filtering that CSF, excess CSF back into the dural venous sinus. So if we have a problem filtering or getting rid of those, then we call this hydrocephalus, a pathologic condition with excess CSF. So we're always making CSF, and our brain only has limited space, or inside the skull there's only limited space. So if we didn't recycle it, if we didn't also absorb CSF, then we would start building too much. And so if we have obstruction in that CSF resorption, then we can cause a problem. So we have a buildup of too much CSF. In a young child, where their sutures are still open, right, the fontanelles are still open, the whole head can actually get bigger because of so much pressure there. And so this can be treated surgically. You can implant shunts that drain the CSF to other body regions. This is the looking down on the brain, anterior, posterior. So we can see here that this is going to be the cerebrum. We have this fissure right here that is dividing the brain into right and left hemispheres. So it's called the longitudinal fissure right here. And it's a pretty deep fissure, and that's kind of, you should be able to pull the hemispheres apart a little bit. We can see in the front, these are the frontal lobes. So you have one in each hemisphere, the parietal lobes, one in each hemisphere, and the occipital lobes are in the back, one in each hemisphere. And the temporal lobe you cannot see on that picture because it's a little bit lower. We'll see it on the lateral view in just a second. And the insular lobe is not visible at the surface, but I'll show you where it is in just a bit. So the frontal lobe is going to be this guy right here, and it is going to come up here to this central sulcus, so that's one you need to know. And this gyrus that's right in front of the central sulcus is called the precentral gyrus. And we'll talk about this in lecture. It's where we have our primary motor cortex, but we'll need to identify the precentral gyrus. And so this is the frontal lobe. The parietal lobe is right behind it, and it's going to be right behind the central sulcus. And so this parietal lobe, this first gyrus here is called the postcentral gyrus. So it's right next to the central sulcus. The functions, we're not going to talk too much, but the functions of the frontal lobe are mostly motor, as well as like decision-making, planning, and things like that. Whereas the functions of the parietal lobe are going to be somatosensation, so like body sensation, pain, pressure, and sense of touch. The temporal lobe is going to be right below the parietal and frontal lobes. And so, here's a good picture. Let me go way back up here. Sorry. Didn't realize there wasn't a good picture of where the temporal lobe is. So this is the temporal lobe right here. So we've got frontal up here, then parietal, and now temporal. So the temporal lobe is going to be right below this lateral sulcus. And the temporal lobe has both auditory, so hearing functions, as well as smelling functions. Now the insular lobe is actually going to be right here. So if we opened up the lateral sulcus, and you can do this on some of the models, and there's some pictures and some videos that you can see that. Then the insular lobe is going to be right under deep to this lateral sulcus. In the back here, we have the occipital lobe. The occipital lobe is going to be primarily, back to the insular lobe, it has to do with taste. Now the occipital lobe has to do with vision. Those are kind of the main things you need to know for the cerebrum. Remember that the sulci are going to be the valleys, and the gyrus are the hills, or the gyri are the hills. All right, so here's the lobes. Now moving on to the diencephalon, this is the middle part of the brain, and the diencephalon is going to include everything that has thalamus in the name. So the epithalamus, the thalamus, and the hypothalamus. And so the epithalamus, the main thing to know about the epithalamus is the pineal gland. So that's this little guy right here, and that's going to secrete melatonin, which is important for circadian rhythms. The thalamus, we can see it's kind of this dark area here, this brownish area. This is the interthalamic adhesion, so it's going to kind of pierce through the third ventricle, and you actually have the thalamus is really big, but you can't see much on midline because in midline, it only has that tiny little interthalamic adhesion. So there's actually some big thalamic bodies on either side. We just can't see it as well on midline. This is sort of part of it. The third ventricle is right on midline, so this area here that's outlined in purple is where the third ventricle would be. So we would kind of be cutting through the area where the third ventricle is. Now the hypothalamus is down here. So the hypothalamus is below the thalamus, and it's going to be continuous. It has the stalk, it's called the infundibulum, and it's attached up to the pituitary gland. So this hypothalamus is going to be, has lots of functions, and we'll talk more about those in lecture. So this part I said, remember that the thalamus actually has is really large. We just can't see it as well on that that mid-sagittal view. And the interthalamic adhesion is where these two large lobes are going to be attached. You don't need to be able to know all of these different parts, just be able to say thalamus. The brainstem is going to connect the cerebrum, diencephalon, and cerebellum to the spinal cord. It contains both ascending sensory tracts and descending motor tracts. So these are like axons going to the brain or away from the brain. And we'll talk more about that with the spinal cord. It contains a lot of autonomic nuclei. So these nuclei are going to be groups of brain cells, groups of cell bodies of the autonomic nervous system. It also has groups of cell bodies that are connected to cranial nerves. And we'll learn about cranial nerves next lab or so. And some reflex centers, mostly for homeostasis maintaining. The midbrain, pons, and medulla are the three parts that you need to know. So the thalamus is up here. So you have a model that looks just like this. The thalamus is on top. Below that is the midbrain. And these mammillary bodies right here, these actually are extensions that kind of come out of the side of the hypothalamus, but we can see them right here on the midbrain. So there's paired mammillary bodies. Right above it is going to be the infundibulum. So this would be connected to the pituitary gland. So those are things to be able to identify. This part right here is the pons, and then this is the medulla oblongata. And these two things right on the anterior surface of the medulla oblongata are the pyramids. And right here, the decussation of the pyramids is lots. Remember, or we will mention in lecture, that the left side of the brain is going to control the right side of the body, and the right side of the brain controls the left side of the body. So most of these motor tracks cross, and right here is where a lot of these axons cross at the decussation of the pyramids. Now, if we look at the back, so this was the anterior view, this is going to be the posterior lateral view, so this is the back. We can see that this part right here is the thalamus is still up here. This is the pineal gland, and we saw that sort of on midline. Now, these guys right here, this is the tectile plate, or the corpora quadrigemina as a whole. So the top two are the superior colliculi, and the bottom two are the inferior colliculi. You could just call them the tectile plate. That'll probably be the easiest thing, and we'll mention a little bit more about their functions in lecture. So this is kind of the back part of the pons, and then here's the back part of the medulla oblongata. So those are the main things to be able to identify for So the cerebellum is going to be that little tiny brain. It's at the back. It's going to be, just like the cerebrum, it has an outer cortex of gray matter and an inner sort of white matter. And it also has right and left cerebellar hemispheres. And it's going to be in between, so at the back of your head, you have the cerebellum and cerebrum, and there's a transverse fissure that goes right in between them that is going to be connecting them. So the major regions that you'll need to be able to identify are the cerebellar cortex, which is the outer gray matter, and the arbor vitae, which is the internal white matter. So this is right here, this is the cerebellar cortex. So this is the gray matter on the outside, and then this white part right here that looks like a tree, or the tree of life, the arbor vitae, this is the white matter. And you can see the two different cerebellar hemispheres. You don't really need to know about the lobes or the vermis. Right. So I'm going to really quickly go into APR and into your down here to see a couple other things that you can look at. You can practice labeling the brain. There are these really cool widgets that Dr. Yates made. So this is a little widget of the meninges that you guys can look at that's labeled. You've also got a brain widget that you guys can look at and labeled, so you can kind of move them around. This is cool since we don't have the actual hands-on models right now. And so you can turn it around, move it around, and here, let me move this. You can see that there are a bunch of different options. You can show labels, hide labels. You can open the Explorer tools. You can fade or hide different pieces. So you can kind of look and see and move things around. You can see the ventricles, you can see different areas. So this is a pretty cool little thing to look at, just a little widget to play with. This is just a GIF of the ventricles that you guys can look at. That's where the cerebrospinal fluid would be inside the brain. And then these are going to be really helpful, these pictures right here, labeling your brain models. So you can see these are the brain models we would have looked at in class. And I've got lots of videos of brains that you guys can look at down here, as well as some of the different lobes here. So I should hopefully have gone through mostly everything. I do want to go into your APR revealed. Oh, got to relog in. So if you go into APR revealed, there's a couple of things you guys can look at. So if you go to the nervous module here, maybe. Hello. There we go. All right, so we're going to go to module, we're going to go to nervous. And so now for here, you would look at brain. And you can look at the lateral view, maybe the brain. And so I usually like to just go through and click the different layers so we can see, oh, this is the dura mater on top of the brain. And then we can go here and we can see the brain and you can see the different cerebral hemispheres. It's working really slow today. The lateral sulcus. We can see the deep parts of the brain in here too. The midbrain, cerebral aqueduct, third ventricle, thalamus. Usually when you highlight it and click it, yeah, there we go. It should highlight. It's just really slow today. So you can look through this. This will be really helpful, I think. And it also tells you the location, the function. The other thing you can do is now the new thing they've added is regional anatomy. And so they've added some regional anatomy and go to all content. Make sure, since I have not customized any of this yet, it's pretty new. And I think that the only regional anatomy they have so far is the head and neck. But either way, that's where the brain is located, so we can look at that there. This one it's like a 3D view, so we can look all around at it. And because of that, it takes a little longer to load. So we're going to be looking mostly at the nervous system. So you can say this right here, this is visible, this is like transparent, and this is gone. So you can make things transparent. Well, not the nervous system. So you could make everything transparent if you wanted to. I'm going to say let's just get rid of the muscles and let's get rid of the cardiovascular. And if we want, we can make the skeletal transparent just so we can kind of, you know, keep our orientation here. So we can look through and you can move the brain around and look at it. If you open up the nervous system here, we can talk about the lobes of the cerebrum. So we can find the frontal lobe, move it around, the occipital lobe, move it around, parietal, temporal. So that's really neat, I think. You can see a lot right there. The cerebellum, we can see the thalamus. We can't see the thalamus very well. So let's go through and let's make the lobes of the cerebrum. Let's get rid of the bones. We'll get rid of this. And so now we can just look here. So this is just going to be the thalamus and the brainstem. We can also see, so here's the thalamus. And let's see, somewhere, hypothalamus. And we might have to look on midline. Let's see. View, anterior, posterior. All right, well, we can move the view around. I'm going to turn the spinal cord off for now because we don't need that. All right, so the ventricles. I don't think it's highlighting properly. I'm going to turn the ventricles off, but we should be able to see. So the amygdala, oh, here it is, it's highlighting now. Amygdala is one that you guys, we talk about. The mammillary bodies, it should be, yep, they're right in there, the mammillary bodies. Let's see, the hypothalamus, it should be right above there. The hypothalamus is going to be on midline, you can't see it very well. Oh, I think it's might be highlighting in there. That should be the pineal gland, which would be the brainstem and endocrine gland. So that's the pineal gland back there. So this should be your tectile plate somewhere. It's in the midbrain, so your tectile plate is back there. So there's a lot of cool stuff that you can see on here. Your cranial nerves, I'm going to turn them off right now. We are going to be looking at the cranial nerves, but not in this lab. And we can also zoom in, we can zoom out. So this, I think, is really pretty cool. Let's see, we can remove the thalamus, maybe, and we can go through here, the brainstem. Oh, we can make the brainstem a little bit see-through. So you can do a lot of cool stuff in here. I do think that it's really cool that you can kind of see how the ventricles wrap around the brainstem, and that kind of helps a lot. So this one is not as useful because I don't really think I can see a way to look at it from the inside, but we can definitely see a lot from the outside. And I haven't played with this enough to really see if I can view, if I could cut it on like a sagittal section. But we can go back and add everything back, and then you can see like a whole bunch of the head. So that's a pretty cool one we can look through. So don't forget to use APR. APR is a really good resource, especially since we don't have access to the models right now, since we're quarantined.